So today we had Dr. Bo Hu from the Department of Bioproducts and Biosystems Engineering scheduled to give a talk about their work on nutrient removal and recycling of nutrients, the nutrients being removed from agricultural waste. And but he wasn't able to be here because of some prior commitments. So one of his postdocs, Bradley, is going to be giving the talk. He received his PhD in 2013, has been in postdoc here for a few years, and he told me just a few minutes ago that he's going to start a new position as an assistant professor in China in June, and be working on very similar things that he's been doing here with energy efficiency in rural areas. So with that, we'll let Brad get started.
Well, thank you, John, for a nice introduction. So I will give this talk on behalf of Bo Hu, and today is the commencement day. And so the topic is process development for nutrient removal and recovery for agriculture and rural wastewater streams. There are some animations. So we talked about the nutrients pollution from the agriculture with a lot, and in a lot of cases, this has something to do with the concentrated animal feeding and operation systems that generate a huge amount of animal manure, and then it's land applied, agriculture runoff goes to the nearby environment, either groundwater or surface water body. And in reality, when farmers are applying the animal waste on crop land, they are applying it based on the nitrogen requirement for either corn and sometimes soybean. And they will find phosphorus in excess. And then it goes to aqueous environment, create some contamination issues. Some of you may heard about dead zones in Gulf of Mexico because of nutrient overloading. And some of you may heard about Toledo, that's a city in Ohio, and there's a drinking water crisis because of the algae contamination. And some people figured out it's most likely because of the swine operations nearby the lake in the city. So there are some problems we can solve as agriculture engineer. Another case we are interested in is the rural household wastewater treatment. So in the United States, about 25% of the households are relying on septic system to treat the wastewater. And according to EPA's estimation, they said that about 10 to 20% of septic systems actually malfunctions. They failed, and a lot of nutrients overload to rivers. That's not only happened in the U.S., but also in Britain. And some people investigate the water quality through the catchment in the river, and they found like 20% of the septic systems around the area malfunctions, create either bacteria, fungi, viruses, and nutrient contaminations to the river in Britain. So two important issues. Sorry, I have to take off my coat. Yeah. So in this research group led by Dr. Bo Hu, people are interested in basically 3 different processes, working on different waste streams. The first is the microalgae co-culture or algae fungi biofilm, and the second is the microbial electrochemical based processes, and the third is the modification of corn ethanol co-products. All of them are targeted on the nutrients management. First case study, microalgae fungi lichen biofilm and its application. The first step, I think, is screening the phosphorus accumulating fungi. When Bo came to UMM, he was really interested to remove phosphorus from the water environment. So he sent students, or he probably traveled together with his students and take some soil samples from soybean crop, soybean leaf, and the soil, and then trying to isolate some fungi species that could accumulate phosphorus in the fungi hyphae or inside of the cell. Why use the filamentous fungi rather than bacteria? It has some benefits over the bacteria. As you can see, fungi produce a group of enzymes that could help to dissolve organic matter, and that is important in the wastewater treatment process, because if you want to remove the suspended solid from water, if those solids are solubilized, then that's okay. And the second, fungi has a greater resistance to inhibitory compounds, like some people mentioned the pesticides. Fungi will sustain pretty well when the soil water is contaminated by pesticides. And the third, we thought the fungi has better adaptation under low nutrient condition than bacteria. I'm not a biologist, and I don't know. I can't explain why that's the case. So, in the literature, based on the rationale I just mentioned, actually, we found a lot of different mucus species or spatulus. They are able to, accumulate the phosphorus up to like 6 or 7% of the total dry biomass. So 6 or 7% of phosphorus in the total cell dry biomass. That's pretty good, because generally bacteria, fungi, or other plants, animals has 1% of the dry mass as a phosphorus. Those fungi shows potential to be used for phosphorus accumulation. So the group tested 70 strains, and eventually there were 9 strains stood out, and some of them could remove up to 86% of phosphorus in the medium cultivation, and some of those fungi species are able to accumulate 6% of phosphorus in the cell biomass. Those 2 parameters are important because first, we want to remove phosphorus from the liquid phase, right? And the second, we want to improve the phosphorus concentration in cell biomass, because that means we can recycle the phosphorus in it for some other application purposes. So there are some other parameters the students tested, including the total nitrogen removal and the COD removal. If you look at the horizontal bars on the right-hand side bar chart, you'll see the phosphorus removal, the blue bars are usually very high, but for the nitrogen, it's usually between 20%. That needs some improvement, right? For the COD, it could reach 80% or 90%, pretty good. This is growth curve for a specific mucus species. When it grows, it uptakes phosphorus in the cell, and then the cell could retain the phosphorus inside of the membrane, inside of the cell for a pretty long time. As you can see, from day 2, the dry biomass, the phosphorus removal has reached, the concentration has been reduced to like 4 millimole per liter, and even after another 5 days, the concentration is still around 2. That means the fungi are pretty good to remove phosphorus and retain phosphorus in their cell biomass. The mechanism, is most likely based on the polyphosphate granules formation inside of fungi. This is a test used to indicate to illustrate the polyphosphate granules inside membrane. Any of you, working on the enhanced biological phosphorus removal process, you probably heard about the polyphosphate accumulation. So EBPR is widely used in the municipal wastewater treatment, in the centralized wastewater treatment. So first step uses activity sludge to remove the COD, and the second and third step you use the combined process called biological nutrient removal process to remove phosphate and nitrogen. One mechanism to remove phosphate is through the polyphosphate accumulation inside of the bacteria cell. And interesting happens the same in the fungi species for testing. As you can see, the dark area indicates the polyphosphate granules inside. And the second step, he's interested to see, where, if we apply some phosphorus fertilizer in cropland, there are some symbiotic relationship between fungi, bacteria, and plants. If the phosphate fertilizer is not so soluble, is that possible? The plant could utilize those phosphorus anyway. So he trying to secrete some phosphate solubilizing fungi in the second step of study. This is called Picavuscaria agar, and it's a plate. There's some calcium phosphate agar dispersed in the medium as a colloid system, so it's opaque before fungi grow. And the fungi grow, when fungi grow and they will solubilize the calcium phosphate, it becomes transparent. So that there's a circle, transparent circle, so we can measure how a fungi species is able to solubilize calcium phosphate. Yeah, here's an animation here. Okay. First circle, second circle, transparent. You can measure the diameter of both, and then you can define the index. That's called a solubilizing index, SI. The second, approach that developed is using the iron phosphate rather than calcium phosphate. In Minnesota, when we visited some local farmers, talk about to dairy farmers or swine farmers, they grow corn and soybeans. They mentioned in their cropland, they usually detect a very high concentration of phosphorus, but crops seem not able to utilize those phosphorus. And some people think it's probably because of the high concentration of iron in soil that we bind with phosphate and make it insoluble. Right? So this is the second medium we used. And the strains that are able to solubilize in phosphorus are mostly trichoderma fungi species, sometimes aspergillus species. But you see, most of them are trichoderma with a very high SI. The highest one, the last one, trichoderma species is not identified yet. The SI index is 3 times higher than the other species. Very good to solubilize calcium phosphate. And also this species was found very good to solubilize iron phosphate. So Bohr is developing a process that he could apply one or two or combination of these fungi on cropland and see if phosphate will be solubilized from the cropland for corn growth. As I mentioned, the first interest was to screen phosphorus accumulating species, and it can be applied for the synthetic, applied for the sewage treatment or wastewater treatment. Both students conducted a separate experiment. I mentioned before, this mucus species is good for phosphorus removal, good for COD, but not good for nitrogen. The same thing happened for not only sewage, but also in swine manure. Remove phosphorus, but not nitrogen. What can we do to improve that? This is a process called cell pelletization by fungi. When fungi grow inside of a flask, when shaped, there will be some pellets formed because of something, and then for optimize the co-pelletization process to form as many pellets as possible to make the surface area of pellets very large. So there are some potential applications for these pellets. Here is one application cases for developed. It's called fungi co-pelletization to assist algae harvest. Industrial microalgae productions, in a very large pond or sometimes in lake. And when algae grow, people need to harvest them from the huge amount of water. How to do that? The algae can be used for a lot of applications: protein extraction, oil production, biodiesel production. But first thing first, you have to get them out, concentrated algae. Gravity sedimentation. It's cost effective, but it's a very slow process. Centrifuge, it goes very quick, but the cost is very high. And eventually, both thoughts, pelletization or co-pelletization through the fungi and algae will probably help the harvesting process, because it has a lot of benefits. Easy to harvest when it's co-pelletized together. It will basically settle down on the bottom of the container, and then you can pump them out. And the second, after co-pelletization, it decreases the viscosity, so cell in medium or in the water can grow faster and has a smaller mass transfer coefficient for nutrients and products. And the third, it decreases the mutual shading effect and allows sunlight to penetrate for higher microalgae production and accumulation. It's understandable, right? If the algae are dispersed in the bulky medium, it shades sunlight. When it's harvested by co-pelletization, sunlight penetrates, so the energy utilization efficiency can be improved. These are some pictures that shows the co-pelletization of microalgae and fungi. There's algae medium, and then you add fungi pellets inside of the medium in flask. And then it will absorb those algae, and you see the liquid is very clean after the co-pelletization. Some tests basically remove the majority of algae. Most time, over 50% in the pellet surface. That's good. Different strains. The next question is, you cannot always, develop the fungi pellets and algae and then put them together. This is the process we call the autotrophic or heterotrophic conditions for the co-pelletization. You grow algae and fungi together and see how the removal proceeds. And the result shows that's also very good. Remove 6% of algae biomass from the algae liquid. This is an example. Autotrophic condition, algae in the suspension, this is the sublatent. This is the total mass. So you can compare this portion and this portion. You will see 6% of the algae cell biomass are removed by the pellets co-pelletization process, either in autotrophic or heterotrophic process. Okay. The issue with that is both identifies the removal efficiency for the phosphorus and the nitrogen most likely are around the 60% or 70%, and an improvement is needed. He observed the natural systems. He observed together with Ara to see, is there some improvement he could do, and then he see a concept called symbiotic biofilm, and in the natural system it's called lichen. Inspiration from the natural systems. natural lichen system. It includes algae, includes fungi, and they grow together on some supporting surface. With that observation, Bo and Ara developed biofilm on some texture, and we thought there are two benefits. First, the algae will, utilize the sunlight to produce oxygen, so you don't need air in the fungi cultivation process. And second, we thought the fungi will grow, and so it will produce carbon dioxide, and the carbon dioxide is a substrate for the autotrophic growth of algae, right? With that, they developed, use the three components, texture, fungi, algae, put them together, and exciting things happened. As you can see, this is a mature lichen biofilm. Fungi grow on the texture and then absorb the algae together, makes it a very dark green color stuff. And consequently, the liquid or the supernatant becomes very clear. Again, it can be used for water treatment, either in sewage, dairy, swine, manure, supernatant, or manure supernatant, agricultural runoff, septic tank effluent, or septic sewage. Observations is through the microscopic. As I said, carbon dioxide, oxygen, they produce these two gases to mutually benefit each other on the surface. And then the research group tried to optimize those processes for algae harvest and for nutrient removal. The parameters evaluated include the matrix type. They use the metal cotton texture made from cotton polypropylene, agitation intensity, temperature between 27 to 35 degrees C, and different nutrients concentration of glucose, nitrogen sources, phosphate sources, and different fungi, algae species. Combine them together, just like cooking, adding different vegetables and meat, and then see how it tastes. Right? It's a process, development process. At the suitable condition, we could achieve almost 100% of algae harvesting efficiency. This also confirmed when the algae is utilized, it not remove phosphorus efficiently, which is 6% as the maximum, and when it's cultured together through the biofilm, it removes close to 100%. Same thing for the that's for phosphorus. Same thing for the nitrogen. This is the algae. Is fungi is able to remove 20% nitrogen. When put them together, and they could remove nitrogen as high as 90%. This is one case study for the algae fungi co-culture and biofilm formation. So they developed this concept and collaborated with Bruce Wilson for floating island test. They designed a floating island or some contact, biofilm contact, and installed them in one, I think, is in lake. It's called Sarita Lake, close to the state fairground. And another place they tested in some solid waste landfill lichen. It's a pond. They put those. They will put a contact over there to see the removal efficiency. I think the research group and Bo is also open for more collaboration opportunities when there are some benefits can be achieved by this concept. Systems that microalgae biomass needs to be harvested. Systems that a single microalgae fungi strain cannot fulfill the purposes of water treatment, and a system that aeration is needed, and that is a very costly process in wastewater treatment from the activity sludge aeration for the activity sludge process consumes 30 to 40% of the total energy consumption. And the second technology platform for developed is through the microbial electrochemical systems. And again, it's utilized for nutrients recovery from different agricultural or rural wastewater streams. Then again, I have to mention the septic tank. As I mentioned, 10 to 20% of U.S. septic systems, or some people call it subsurface wastewater treatment systems, on-site wastewater treatment processes. 10% of malfunctions, and we need some, improvement. And some of you may heard about garbage grinder or garbage disposer, food waste disposer, whatever name it is. 50% of U.S. households install those food waste grinder, and we also evaluate how those grinders affected the water quality before and after the septic tank treatment. Several things going on. The first is the food grinder, food waste and sewage degradation in septic tank. So generally, literature indicates when the household process the food waste in wastewater stream or septic tank, there's about 30% of total suspended solid or COD or BOD increase. And with that increased load, is like septic tank able to treat this additional waste? Is wastewater treatment plant able to treat this additional load of waste? It's questionable, and no one can answer the question. So this is the first study about the septic tank, evaluate what happened when food waste was spiked with the sewage. Basically, we found food waste is easier biodegradable than sewage solid. You can see from this biogas production. The red lines indicate the biogas production from like 30% of COD concentration of the combined substrate. And the blue line indicates the biogas methane production from the 70% of the organic matter from sewage solids. So one is 30%, but it produces way more methane than the other 70% of sludge. So that indicates that food waste is either degradable when you flush food waste to septic tank. It's not going to increase the total suspended solids in the proportional ways in the substrate, because a lot of them will degrade. And then we monitor the water quality in the influent effluent to see what happened. And we see the gradual mature of the septic tank. And even after like 209 days, the septic tank is improving itself in terms of the COD, including total COD and the soluble COD. Amazing thing. Total COD, soluble COD. Let's move to the conclusion part very quickly. Okay, this is the most important slide here. So the first two lines are the sewage solids, and this is the last two lines are the food waste solids. 50% of the sewage solids accumulate eventually in tank. Only 20% of solids from food waste accumulate. And why? The major reason is the food waste solids are easily biodegradable compared to the sewage solids. One is 6%, the other is 40%. And the second thing is, let's take a look at. Okay, in the septic systems, the phosphorus is removed in the leaching field, right, through the soil absorption process. You cannot remove phosphorus inside of the septic tank. Only the soil treatment system doesn't. So we are thinking about, can we remove phosphorus before the effluent goes to the soil filtration system? So we use the electrochemical. We insert the electrodes in septic tank. And we did two experiments. First is small scale, and the second is a large scale demonstration in simple municipal wastewater treatment plant. Some mechanisms for phosphorus removal, some species validated for the elemental composition, magnesium, calcium, phosphate, iron, sometimes. Configuration, different conditions. Okay, here we go. Phosphorus. Let's see what is the phosphorus removal. At the two temperatures, we tested the two temperatures, 25 degrees C and 15 degrees C. That's the highest removal to achieve the 99%. There's basically no phosphorus, less than the type of phosphorus remain in the effort. So the soil will never saturated for phosphorus removal. If we can apply this concept for septic systems. The sludge volume is increasing with electrochemical processing inside. That's the worst case is the volume increased by twofold. And methane production improved residential chemical systems. And comparison with other treatment, we found this process is the feature of this process is so efficient in phosphorus removal compared to other improvements. Some people add iron oxide, ferric oxide, ferrous oxide, more than 10 different species of ferric oxide, hydroxy, iron minerals. None of them could exceed our electrochemical process in terms of phosphorus removal. And then we demonstrate this process in the septic tank. Sewage lifted from the waterway underground to the temporary storage tank, and then goes through the 300-liter pilot-scale septic tank. We collect the effluent in the water in the effluent and effluent, test the water quality every day, except for the weekends. Again, we removed the green line effluent, the black line is effluent, the green line are the effluent. When the electrochemical process occurs, the phosphorus is always removed. When the electrochemical process stopped, but here you can see in the effluent, the phosphorus concentration jumped to like 18 milligrams per liter. It's stored in tank. Phosphorus is stored in tank, and then we did a cost analysis for a process. The funds are too small, but I can tell you what happened. For the conventional septic tank, the roughly installation and maintenance cost, the net present value for the cost is about $8,000. And with the electrochemical process, the insert the electrode power it, the cost is about $11,000. So there's like $3,000 increase. Some people think it's better to use the solar energy to power the electrochemical systems. And we did the analysis. There's like $500 saved using the solar panel rather than the grid power. So it improves the phosphorus removal efficiency, and the cost increases. And some people probably would think it's acceptable to add another $3,000 to the septic tank to improve the process. Swine manure management systems for the odor removal. If some of you go to concentrated animal feeding operation system, it smells really bad. And some companies, they're trying to design modern swine or dairy farm, so people working there could have better environment, and we thought the environment should be as good as university offices, and they're still building, and some people check if that's the true case or not. So currently, in Minnesota, we have found odor issues with manure management systems. Most of the manure management systems in our state is called deep pit management system. They dig 2.5 meter on the ground, store the swine manure inside of the pit, and over the half of 1 year storage, a lot of gases are emitted from the interface, right, including methane, carbon dioxide, hydrogen sulfide, ammonia. Sometimes concentration kind of reach a very dangerous level. I did a simulation, and I actually take some field samples, compare with the literature. This is one we found. In a simulation, when pit is agitated, the headspace in the pit could have a hydrogen sulfide concentration higher than 400 ppm. When the concentration is beyond 100 ppm, it's very dangerous already. And it's called IDHL, immediately dangerous to life and health concentration, 100 ppm. And in the room, when the ventilation is low, it exceeds 100 ppm. Dangerous situation. The results are kind of support the literature studies. This data from a paper I submitted to ASAP, you can see a lot of cases the concentration is 500 ppm, 1000 ppm, not to mention 100. So can we do some improvement to remove hydrogen sulfide from stored swine manure? We did the small scale experiment, and then we are trying to test the larger scale in deep pit. Some people would ask, why you have to do this pilot scale experiment rather than do a simulation? You can simulate the effect of a lot of factors, total solid pH values, volume of manure, the manure increase, the manure increase rate, ammonia concentration, sulfide concentration, a lot of processes. Why you do that? I would tell them, the manure storage system is too complicated to simulate. It's too complicated. You can simulate a bioreactor, simulate how methane can be produced from anaerobic digestion, simulate how biofilter would remove phosphorus and nitrogen, but you cannot simulate the biochemical or biological process in manure storage systems. So we did a pilot scale field test. Morris in West Central Research and Outreach Center, when we visited it sometime in December. We started sometime in January, and we visited a couple times in February and March in Minnesota. Very cold. It was very cold. But eventually we conducted this experiment for 4 months, collected the data, analyzed the data. Okay. Here it is. We use 2 different materials for the low carbon steel electrode. We applied 0.6 volt, 0.7 volt. We monitored the hydrogen sulfide removal efficiency. So we have a control pit. We have an experiment pit. We compare the concentration to get the removal efficiency, 78% removal efficiency. When stainless steel material was used, the removal efficiency is between 13 to 38. So low carbon steel is a better choice for the electrode material. And then we are trying to, design. Can we, apply this technology in a really large scale of animal production systems, 2,400 pigs. We first did an economic analysis, and what we found is when you're trying to install those electrochemical systems and trying to operate them, the cost increase is between 16% to 26%. The data is there, and we will see if some people from the Minnesota National Park Board will be interested in this concept and do a pilot scale testing for a full barn, if not a full barn, maybe a research barn with a couple hundred pigs. And then with this technology installation, we reduce the hydrogen sulfide emission, and we can imagine that sulfur concentration in manure will be increased. And when the petroleum industry remove sulfur from the off gas, a lot of car land they need a sulfur fertilizer. With this sulfur increase, we can contribute to crop production. That's an idea we can test. And also that happened for the phosphate. The phosphorus ratio to nitrogen is high in manure. So use this system, we can accumulate some of the phosphorus on the electrode surface area. And the phosphate concentration on those solid precipitates are higher than the sludge phase. It can be used as a phosphate fertilizer potentially. Another test students done is to use the electrochemical process to remove phosphorus. Again, the purpose is to separate the nitrogen phase, phosphorus phase, and to apply them independently, so you can use a better nutrient management practices by separating them. The results turn very, very promising. The summary slide is not here, but I will tell you, 90% of phosphorus can go to the solid phase. And so the liquid phase is concentrated with nitrogen. How you are going to utilize it, farmers knows better how to utilize it. Apply the liquid and solid depend on nitrogen and phosphorus requirement of the plant. If they grow corn, they probably need both. If they grow soybean, they probably just need to apply the solid. Then the research group is trying to, go along the food chain, as you can see, from animal production to the sewage treatment system, and then go back to the feedstock for the animals, corn ethanol co-products. The purpose again is to, improve the environment by mitigate phosphorus in certain way. This is the general flow of dry ground corn ethanol production processes. And corn grain contains a lot of phytate in kernel, and during the fermentation processes, bacteria are not able to degrade those phytate. It goes to the dried distillers grains and soluble DDGS, called DDGS for the corn industry. Very high phytate concentration, and it's used as an animal feed. Animals are not able to digest the phytate again, so it goes to cropland. As I mentioned, the phosphorus to nitrogen ratio is very high. If we remove those phytate from the feed, then we can decrease the phosphate to nitrogen ratio to improve the agricultural runoff. This is, a patented process both developed to remove phytate from DDGS or corn ethanol intermediate like thin stillage, whole grain, wet whole grain, dry whole grain. And then another idea is to using the yeast, engineer yeast to improve the protein profile. After digestion, maybe the protein in the dry grain is not suitable for animal consumption, right? Can we improve the nutrients by producing more lysine? This is a profile of the nutrients. You can see lysine is deficient, only 0.91%. Lysine, methane, leucine, limiting amino acids, and in the DDGS, we can improve these amino acid concentration. The feed producers don't need to add these amino acids. It's easy for them, for farmers, and they can buy the same feedstock with a lower price. This is a gene modification process packed with preliminary results for lysine engineering. Lysine concentration is increased in the media, in the bulk media, but the productivity of the yeast is reduced. So that means the ethanol production is reduced. There are several factors a researcher has to take care of here. They need to improve the lysine production concentration. They also need to make the ethanol production the same or improve it. So the project is still going on. This is a group member, and the one group member is not in the picture. She's a PhD student, just joined the group in 1 month ago.
You're not.
I'm not in because I'm leaving. Yeah, Aravindan, Laura Zhang, Wang Yishan, and Tanner. 3 of them are in the commencement. Carlos, Bo, Kalis, and P. So, if you have questions about the second part, that is about the electrochemical systems for nutrient removal, I will be able to answer the questions. Otherwise, for the gene modification, lysine production, or for the lichen biofilm formation, Bo is the best person to address your question. That's it. Thank you. Any question? Come on. What research are you doing?
I have a question about it. I mean, all this stuff about phosphorus removal and nitrogen removal. I assume that when you do the phosphorus removal, I mean, the phosphorus is still there somewhere. Yeah. It's been removed, but now it's in a package, some sort of package. It's in the body of the algae or it's in the body of the fungi, right? Yeah. And is that used as a fertilizer, or if you make a biofuel out of the algae or something? I mean, how are you recycling or reusing the phosphorus?
I think that's a question, right? You raised a really important question, and till now we are not able to pursue. Ideally, we remove those, and now we recover them. Yeah. After recover them, we are trying to use them as a fertilizer, but the plant test experiments are very long, and Laura is testing one of the methods using the electrochemical solid, as I mentioned, the phosphate is accumulated on the sludge, and then she applies those sludge on the corn, on the soybean, and see how the phosphate can be used by soybean. Okay. And compare them with the different fertilizer application, different fertilizer types application.
So it's not readily available for the plant.
No. We will see. John, you can probably after like 2 or 3 rounds, you can talk to Bo and you probably know the answer. They have like a 5 triplicates for each think the results probably will be very, they can reduce the variation by using 5 replicators, right? And they control the light system, randomly assignment. So I think it's a good experiment design. We should see the results. Ideally, we better to utilize those phosphorus as a fertilizer, either as a true white or iron phosphate materials applied on the cropland. If the cropland is not able to solubilize them, it's better to apply them on the lawn. So our neighborhood could utilize it. I think that's the important question. A lot of people are interested in. Unfortunately, we are not able to. Yeah, go ahead.
Those are sludge used as fertilizers. Those contain some pollutants or some contaminants.
What contaminants are in your mind? What kind of contaminants?
I don't know, like heavy metals, some contaminants.
That probably could happen, especially in the dairy operations. The cow dairies, there's a rotten food. They have a rotten food. It's a disease. So they have to walk the copper solution from time to time. So the copper concentration in the liquid could be high. And when electrochemical process is applied, it's possible the carbon could be concentrated in the solid as well. I think the same thing could happen for even algae fungi co-cultivation, because fungi are able to absorb a lot of metals on the surface. So that's a concern. But just like the issue we have for the manure application, after years and years of application, we see some metal concentration increase in the soil. But we have no choice. We have to still apply the animal manure on cropland as a fertilizer or as a waste management process. For those recovery technologies, still now no one ever tested them. But I think your concerns are pretty valid. Some research are needed to address the issue. Thank you.
I was sent a proposal from actually Patrick Cameron asking about microfiltration. They were using fungi, so they call it fungal filtration for, let's say, wastewater or urban stormwater, trying to take out heavy metals, E. coli, and some nutrients. Now, I'm not sure if yours is what you've been working with is considered filtration or if it's considered processing that would take a longer residence time. How would this relate?
I think they developed a separate concept for the fungal biomass. The first, they think it's electrostatic interaction between fungi and other things. So they use something called a zeta potential to detect the electrostatic condition of the surface. And that process could be used to explain the interaction between fungi and algae. They actually attract together with each other. And something not mentioned here is heavy metal. I think that probably is through some other processes rather than the electrostatic interaction. I'm not sure. But it's there are so many studies about heavy metal removal by fungi biomass. And I think in the literature, maybe some people have validated already either through the mentioned phosphate binding with some heavy metals on the surface. If the fungi are able to grow to attract a lot of phosphate on the surface, or maybe some polyphosphate, it will have residues anyway. The residues of phosphate will bind with metal on the surface, or maybe through check the colloid systems to see if some dimensions less than 10 nanometers will play a role in it. I'm not familiar with that, but I think there are some studies. But of course, it depends on the fungal species. For the microfiltration through new studies for the E. coli removal, I think that it could happen because of one of the students, Christiana, he graduated like 1 year ago. He prepared some fungi biomass filter, and he can remove most suspended solids from thin spinach. Do you have his email? I think the last page is this one, BHU. What's your question?
So when you have the yarn matrix and the I guess co-pelletization, after you use the algae or lichen filter, removed that from matrix, what do the yarn reuse, or is it just trash?
So in this test, our Vincent did is he was using a matrix made from cotton and polypropylene. It doesn't degrade in our stomach. It's harder to degrade in the natural environment. So if you are able to find some material that's degradable by animals or biodegradable to bacteria or fish, you can put them together. If there are no toxins, you can use as a feed. One of the both proposals is trying to use that for fish feedstock. But of course, they need to replace the polypropylene from the yarn by other biodegradable stuff.
Yeah, I would imagine it would break down in the water or in the sludge.
But your idea is brilliant.
Thanks.
Okay, both email is over there. Yeah, send him.
Thank Brad.
Okay, thank you. It's commencement time, and I think they will have some food over there. Okay.
First,